Hi, my name is Nate Smith, and today I'm going to be talking about some of my dissertation work focused on the foraging behavior of the desert leafcutter ant, Acromyrmex versicolor. I'm interested in how animals choose what to eat, and because feeding is such a fundamental part of biology, this is a question which biologists have been studying for many decades. I'm especially interested in a relatively recent area of research which focuses on how the relative concentration of specific nutrients in food items influences foraging decisions. Foraging decisions become increasingly complex for animals that live in social groups that involve division of labor and sharing of food among individuals with differing nutritional needs. They become even more complex yet again in an agricultural species like the focal group of my dissertation research, leafcutter ants. Leafcutter ants live in one of nature's great obligate mutualisms. The ants provide food, primarily in the form of freshly cut leaf fragments, to a fungal symbiont, which in turn produces nutritionally dense hyphal swellings that are consumed by the ants. The focal species of my dissertation work is Acromyrmex versicolor, one of North America's two native leafcutter species. I analyze my nutritional data using the geometric framework. This is a graphical approach to understanding data, and it revolves around nutrient spaces. A nutrient space is formed anytime two nutrients of interest are plotted against one another. Within a nutrient space, a food item is represented by a line whose slope is equal to the relative concentration of the two nutrients in that food. A key concept in the geometric framework is that many animals defend what is called an intake target, which represents the optimal intake of the two nutrients of interest. An animal can reach this intake target using a behavior called compensatory feeding. This refers to eating relative amounts of available food items to move through the nutrient space to the intake target. We can use compensatory feeding to experimentally identify intake targets. We do this by offering the study organism an array of different diet pairings. And if the animal changes its relative consumption of the available diets to reach the same point in the nutrient space, then we have identified the intake target. To determine if Acromyrmex versicolor regulates its relative collection of macronutrients, I used a simple choice experiment. I presented the colonies with pairings of experimental diets that varied in their relative protein and carbohydrate content, and every 3 days for 21 days total recorded how much of each diet the colonies had collected. The diets I use in this experiment consist of coarsely ground cornmeal supplemented with either amasoy, a plant protein, or xylose, a common plant sugar. They are also uniquely colored so they could be easily identified. This is a photograph of what one of the colonies looks like when it's undergoing this experimental protocol. There were two main results of this experiment. First, colonies regulate their relative collection of nutrients across a range of colony sizes. On this graph, each point represents the average 3-day nutrient collection of one of the experimental colonies. The dashed blue lines represent the experimental diets, and the red line is the line of best fit for the data points. The colonies used for this experiment varied considerably in size, and while absolute nutrient collection was correlated to colony size, relative nutrient collection was quite consistent. The other main finding was that colonies were relatively consistent in their collection of nutrients across different diet pairings, which is what we would expect if they are defending an intake target. There's a bit of a more nuanced story here that I don't have time to discuss today. It seems like they may more loosely regulate their collection of nutrients than non-agricultural species. But in general, 5 colonies of similar size did modulate their collection of diets based on the relative nutritional content to reach the same area in this nutrient space. During the previous experiment I just overviewed, I'm measuring nutrient collection by the colony as a whole. However, as I discussed in my introduction, this is a socially living organism farming a fungus, meaning that there are ants in different life history stages in the colony, as well as this totally separate species which they are farming. This led me to perform a series of experiments to try and determine how the component parts of this system shape the needs of the colony as a whole. I decided to start by looking at the potential role of brood, or the eggs, larvae, and pupae, in driving relative nutrient collection. I made that choice because in non-agricultural ant species that have been studied, brood play a central role in driving relative nutrient collection of the colony. To study the role of brood in shaping nutritional needs of the colony, I performed an experiment where I randomly assigned 6 colonies into 3 pairs, measured their collection of a pair of experimental diets for 1 week, then moved half of the brood from one of the colonies of each pair into the other colony. I then measured nutrient collection by these colonies with manipulated brood levels for a second week. I used the same experimental setup, with the exception that I collected the diets every day instead of every 3 days. The first time I performed this experiment, I selected colonies based on fungus garden size instead of brood number. So in one pairing, only 12 brood were moved, which was less than I wanted. There were two findings from this experiment. On this graph, the y-axis is the proportion of protein collected. And as you can see, increasing or decreasing brood did not significantly change relative collection of macronutrients. On this graph, the y-axis is the total mass of macronutrients collected. And again, there was no effect of either manipulation. Because of the issue with brood number, the first time I performed this experiment, and also because I was pretty surprised that there was no effect of brood on relative collection of nutrients, I performed this experiment a second time. And once again, I found that there was no effect of either brood manipulation on relative collection of nutrients or on absolute nutrient collection. So it seems that in this system, brood are not playing as central of a role in driving colony nutritional needs as in other non-agricultural ant systems that have been studied. I next moved to studying the role of the fungus garden in shaping nutritional needs of the colony. I used the same experimental setup as for the brood manipulations, except all 6 colonies had approximately 1/3 of their fungus garden removed at the 1-week mark. We couldn't add fungus to colonies as they will reject different strains of fungus than the one that they are currently cultivating. This graph shows that there was a significant decrease in fungus volume when one of the 3 fungus chambers was removed from the colony. It ended up being approximately 27% of the total fungus garden. Once again, there was no effect of fungus removal on the proportion of protein being collected by the ants. However, total mass of macronutrients collected did significantly increase following fungus garden removal. So what this indicates is that the fungus garden may be the main driver of nutrient collection by the colony, and that instead of needing to modulate relative collection of nutrients based on colony composition, the ants just collect more or less food with a similar relative nutrient content based on current fungus garden size. This finding may add another layer to our understanding of the value of fungus farming. I would like to briefly thank a few people for their help on this project. First, Brooke Owen, who assisted me on the colony composition and manipulation experiments to fulfill the requirements of her undergraduate honors thesis. I'd also like to thank Jennifer Fewell and Rebecca Clark, who have both given me a lot of guidance and feedback on these projects, and to thank the rest of my lab for their support. Finally, thank you for your attention, and if you have any questions, I'll do my best to answer them. Thanks.